Hello everyone. Today we are going to read about another topic in cellular biochemistry, that are the cytoskeletal elements. The cytoskeletal elements are the network of protein fibers within the cytoplasm that supports the cell structure, the cell, the organelle movement and cell division. There are mainly three types of filament, that are microfilament, intermediate filaments and microtubule. The microfilaments, the examples include actin and microvilli. They help in the muscle contraction and cytokinesis. The intermediate filaments include vimentin, desmin, and other examples of the intermediate filament includes cytokeratin, lamins, glial fibrillary acidic protein, and neurofilaments. Their predominant function is to maintain the cell structure. Another type of filament includes the microtubule. The examples of the microtubule includes the cilia, flagella, mitotic spindle, and centrioles. Their predominant function is of the movement and cell division. So these are the types of filaments you should know. So two types of filaments you should know in details are microtubule and cilia. The structure of cilia in detail and the microtubule you should know, and I will discuss it in the subsequent lecture. Now let us move towards the microtubule. As I have already said, microtubules examples include cilia, flagella, mitotic spindle, axonal trafficking, and centrioles. Their main predominant function is movement and cell division. So if you look towards the microtubule, it is the cylindrical outer structure composed of helical array of polymerized heterodimers of alpha and beta tubulins. They are cylindrical outer structure composed of the helical array of polymerized heterodimers of alpha and beta tubulin. Each dimer has 2 GTP bound. Each heterodimer has 2 GTP bound. It is incorporated into flagella, cilia, mitotic spindles. It is incorporated into flagella, cilia and mitotic spindles. It grows slowly and collapses quickly. It is also involved in the slow exoplasmic transport in the neurons. If you look at the molecular motor proteins, the transport cellular cargo towards the opposite end of the microtubule. So in the retrograde transport to the microtubule from positive to negative end, the dynein is used, and in the anterograde transport from the negative to positive, kinesin is used. The Clostridium tetani, herpes simplex virus, polio virus and rabies virus use dynein for the retrograde transport to the neural cell body. And the drugs that acts on microtubule are the mebendazole, which is the anti-Alzheimer's drug, the grisefluin, which is antifungal drug, the colchicine, anti-gout drug, vincristine and vinblastine, it is the anti-cancer drug, and paclitaxel also is the anti-cancer drugs. Now let us move towards the cilia structure. Now let us move towards the cilia structure. The cilia structure is arranged as the 9 doublet and 2 singlet arrangement of the microtubules. If you look at like this, there is one arrangement of the microtubule, there is second arrangement of the microtubule, and there is third, fourth, fifth, sixth, seventh, eighth and ninth and so on. Similar structure of 9 doublet arrangement of the microtubule is present, and 2 singlet arrangement of the microtubule is present. In the basal body, at the base of the cilium, below the cell membrane, it consists of the 9 microtubule triplets with no central microtubules. The axonemal dyneins, that is ATPase, that links the periphery of 9 doublets and causes bending of cilium by the differential sliding of the doublets. The gap junction enables the coordinated ciliary movement. Now let us move towards the Kartagener syndrome. The Kartagener syndrome is the primary ciliary dyskinesia. Here the immobile cilia is present. The immobile cilia is due to the dynein defective arm. Dynein arm defect leads to the immobile cilia. It is the autosomal recessive disorder. It results to the decreased male and female fertility due to immobile sperm and dysfunctional fallopian tube cilia respectively. It also leads to increased chance of the ectopic pregnancy. It leads to the bronchiectasis, and other disorders that links to Kartagener syndrome are the recurrent sinusitis, the conductive hearing loss, chronic ear infection, and situs inversus, that is dextrocardia. It can also lead to the dextrocardia. The decreased nasal nitric oxide test. Decreased nasal nitric oxide test is used as the screening test. So this is the example of the cilia and the disorder related to it. This much for today. Thank you.